Right. Hi, everyone. It is my great pleasure that I'm introducing today's Grand Round speaker and a personal mentor to me, Dr. Michael Wilson. Dr. Wilson is the Radcliffe Distinguished Professor of Neurology at the University of California, San Francisco's Weill Institute for Neuroscience and director of the UCSF Meningitis and Encephalitis Center. He got his start, though, in neurology right here as a Partners, now MGB resident, after which he completed a clinical fellowship with Dr. Venna and a postdoc in metagenomic sequencing at UCSF. From there, he launched a career bringing the budding technology of metagenomic sequencing into the prime time, developing a widely available clinical assay revolutionizing testing for neurologic infections. He's also an excellent clinical neurologist. When he attends on the wards at SF General Hospital, his expertise in the complications of HIV and presentations of rare neurologic infections ensure that his patients get the best care. He's a thoughtful and supportive mentor to many, and I count my being here in neurology in large part to his guidance. That it seems like just about everyone in the field of neuro-ID, both in the US and abroad, has had the pleasure of collaborating with him, speaks to just what a joy he is to work with. And his cutting-edge research on translational biomarkers and diagnostic assay development in infectious and autoimmune encephalitis has earned him awards from the Foundation for the NIH, the American Neurological Association, and the International Society for Neuroimmunology, as well as being elected to the American Society for Clinical Investigation and the Association of American Physicians. His lab's newest work focuses on profiling the immune responses to CNS infectious and inflammatory disorders using transcriptomics and antibody discovery, which we are very lucky to be able to hear about more in his grand rounds today. So please join me in welcoming Dr. Michael Wilson.
Thanks so much, Carson. I have two former medical students I worked with from UCSF who are in residence here, and just a real pleasure to see all that you've accomplished. And of course, it's just really touching to be back here. Just have so many wonderful mentors and teachers from residency that really set the stage for me to be able to do what I do. And it's just been a real gift to be able to collaborate with so many people here, both at MGH and at the Brigham. So thank you. Is a really special visit for me. So I will get started. This is my disclosure slide. And then I put another one that's more legible, and I'll just highlight, most of these are not relevant, but I'll highlight, I'm involved with a company, Delp Bio, that's commercialized a metagenomics assay. So I don't touch a lot in this talk on metagenomics a little bit, so that's relevant. And then a couple of antibody biomarkers I'll touch on that here. So I think, a big driver, and this again started through morning reports at the Brigham, I think with Marty Samuels and Alan Roper of, discussing challenging cases from overnight. And of course, I would get very excited when we would discuss an encephalitis case, but also frustrated that even with all the amazing clinical acumen in the room, that we would have these great discussions, and at the end, we'd say, wow, that was a great discussion, but we still have no clue what the hell is going on. And so when I realized that I really wanted to focus on both caring for, but also studying those types of patients in my career, I thought if I have to end, half my discussions with that comment, it's going to be a long career. And so, so that's been a big focus of our work, is trying to chip away at that really stubborn, about 50% idiopathic rate in these various cohorts. It's not all our fault that we do a poor job. There's many reasons for why we fail to make diagnoses in these challenging encephalitis cases. Some, of course, is that even if we had a perfect suite of candidate-based infectious disease diagnostic tests, they can't anticipate new kids on the block or re-emerging infections, which of course is very salient to us the last few years. So I think that's a key reason why we miss cases. And then, of course, this revolution in autoimmune neurology the last 10 to 20 years, and just this kind of ever increasing recognition of the wide variety of autoimmune disorders that we now recognize in retrospect often can mimic what we previously thought were cases of infectious encephalitis. And so the kind of the early, like as Carson said, during postdoc and early faculty work, we did a lot of work developing a broad-based diagnostic assay to look for all possible infections minus prions in spinal fluid. And this has resulted in a number of case reports, case series, and then a larger prospective study from a few years ago, just showing that this type of assay can both identify, totally divergent pathogens, pathogens that are known but just are not linked with the clinical phenotype of the patient in front of you. Rare pathogens that are known to cause encephalitis, but they're so rare that either we just don't think of to test for them, or if we do think to test for them, the testing is very cumbersome to access. Maybe it's only available through the CDC. And then there's a potential role, which I think we're still working on, is if an assay that can look very broadly for infections is negative in a patient for whom you're more suspicious about autoimmune encephalitis, can it help can the negative predictive value of the test really help boost confidence to institute earlier empiric immune suppression? And so just as, one update, this was, we published what was called the PDAID study back in 2019. And then last year, this led by colleague Charles Chu, he reported kind of an update on that work. So in the original study, we did testing on 200 patients. And this is an experience of now about 5,000 tests that were performed at UCSF from 2016 to 2023. A lot of the statistics across the two studies were remarkably similar. One thing that changed was the hit rate. So the assay didn't change, but the proportion of cases in which we identified a pathogen went down from 28 to about 14%. The test stayed the same, but in the PDAID study, we were gatekeepers in terms of evaluating patients and whether they met criteria for being likely to have infectious encephalitis. But after the PDAID study finished, kind of anyone could order the test. And so I think this really reflects kind of our need to still figure out what patient characteristics really should be kind of taken into account for sending a test like this. These were the proportions of, the types of pathogens that the test identified, again, highlighting the kind of broad-based number of infections that can be identified. And just, again, very similar to the original PDAID study, the overall test sensitivity for detecting a pathogen that could also be detected by direct detection in the spinal fluid, so meaning PCR culture or antigen testing, was about 86%. But then the overall sensitivity was 63%, meaning some patients had infections identified elsewhere in the body, either in the blood, abscess fluid, et cetera. So as a single test, it was not surprisingly, it was more sensitive than other types of pathogen-specific assays. And then in the proportion of patients, about over 1,000 that were specifically seen at UCSF, where we had access to the clinical records, about 22% of the infections were diagnosed by MNGS alone. So it really did seem to provide a benefit there. So just moving into kind of other work that we've been doing more with a really wonderful global health collaboration. We've been focused on TB meningitis the last few years. Of course, tuberculosis is a worldwide scourge, and TB meningitis in particular carries a very high mortality, about 50% in HIV-infected patients. And part of that challenge is due to the fact of just diagnosing TB anywhere in the body, but certainly in the central nervous system, it's very, very hard to pick up. The best assay right now is the Gene Expert Ultra, which in the group from University of Minnesota and Uganda has about a 70% sensitivity for picking up TB in the spinal fluid in patients who either have probable or definite TB meningitis. Definite means it's microbiologically proven in the central nervous system. Probable, it could be a patient who's got basilar meningitis and TB elsewhere in the body. So you can make a reasonable conclusion that it's probably TB causing their meningitis, but you haven't proven it. And of course, and in Uganda, these are patients, many of whom have co-infection with HIV. And so there's certainly a possibility that they have a or alternate infection. And so when they say 70% sensitivity, is that because there are many cases that are being missed by the PCR assay? Or is it just that some of those patients don't have TB meningitis in the first place? And so we published a study a couple of years ago where we looked at hundreds of samples from this Ugandan group. Carson Quinn was involved with this, and great colleagues, David Bulwer, David Maya, Prashant Ramachandran, Akshay Ramesh. And the idea was to use sequencing to look broadly for TB, but also or alternate infections, but also to see whether the host response, again, this is an agnostic RNA sequencing tool, so a lot of the data that comes out of this infectious disease test is actually human gene expression data. Could we use that part of the data set to help better classify patients? And so again, we looked at nearly 400 spinal fluid samples, and the quick upshot here was that we did find TB in about 3/4 of the cases where we knew we should. But we also found a lot of other organisms. So we found co-infections with toxoplasmosis, Cryptococcus, varicella zoster, even in cases that either had definite or probable TBM. And then, as you can imagine, the patients who had possible TBM, there was a whole slew of other infections that I won't touch on here because I want to focus on the host work. So we were just happy to see that, even though we only have a few picograms of RNA in the spinal fluid, we were able to recover a pretty robust transcriptomic data set. So we got non-zero transcript counts for about 3,000 protein coding genes per sample on average. And using that transcriptomic data, Akshaya used a variety of machine learning models to develop a classifier to just discriminate between patients who either did or did not have TBM. They had some other documented infection. And so she trained this classifier, and just as a first kind of sanity check, we were happy that the kind of top 4 genes that were most predictive of TB meningitis all had kind of a TB-associated literature with them. In fact, this top most explanatory gene, GBP5, is actually part of a 3-gene blood-based classifier to diagnose TB. So that was kind of a first sanity check. But then in a blinded test cohort, we took that classifier and evaluated another 130 samples to see, just based on the host response, not on pathogen detection, could we accurately call people as either having TB or something else. And this blue dotted line is the accuracy for just the host-based classifier. And then if we layer on top of that the enhanced specificity from direct pathogen detection by metagenomics, we got an overall sensitivity and specificity in the high 80s. So we were happy about that. And that, again, that's been published. But more recently, we've been trying to look at the host response within patients for whom we really are confident that they have TBM, to see whether there are any correlates of survival there. Why do we think about that? Well, there's been a long debate in the literature about whether we should give steroids concurrently for patients with TB meningitis, because there's good belief that the bug is harmful, but the immune response can be more harmful in some cases. And so there was a clinical trial, randomized trial published from the Vietnam group 20 years ago, suggesting that adjunctive dexamethasone did provide a survival advantage for patients with TBM. But about 20% of their patients, so about 100 of the patients had HIV. And in that subgroup, albeit an underpowered one, they didn't see a benefit. So that led to a second study, which was just published 2 years ago now, basically replicating the first study, but doing it in an all HIV population, so about 500 patients. And these are patients with either possible, probable, or definite TBM. And the upshot was that in this population, they couldn't show clear-cut survival benefit with steroids, which stands to reason. They didn't just have HIV, they had very advanced HIV. And it stands to reason that in an already immune-suppressed population, adding on adjunctive steroids on top in the face of a potentially fatal infection could be harmful. So we wanted to understand this finding a bit better, and so led by a former fellow, now Martino Luine, we kind of sequenced more cases and first whittled them down to, after looking for or alternate infections, tried to whittle it down to cases for which people either had definite TBM without a significant co-infection like toxoplasmosis, and also looked at the probable and possible groups, and really tried to hone in on patients with probable or definite TBM for whom we didn't find an alternate or co-infection by MNGS. And so that led us to about 100 cases that were demographically quite similar between folks who did survive in the short term versus those who did not. This isn't projecting well, but there is a volcano plot there, I promise. And this is basically, we did differential gene expression to look at what genes were differentially expressed in the short-term survivors versus not. And again, thankfully, we were able to get a robust transcriptomic data set from these samples. And the upshot was, and this hopefully going to be submitted soon, was that we, consistent with the clinical trial data, in this kind of more curated cohort of patients, found that the survivors had a number of, especially T cell signaling pathways that were upregulated, as well as tumor necrosis factors, so innate immune pathways, compared to folks who didn't survive in the short term. So I think, in addition to those specific findings, I think this highlights to me that for additional TBM trials that are out there, I think at adding on MNGS to to improve patient selection, to avoid having people in TB meningitis trials who don't actually have TB is important. And then I should say on the side that these using the host transcriptomic data to differentiate between disease states is something that we're also working on with domestic cohorts. Of course, not TB versus other, but we'd love to develop a classifier for instance, for viral versus autoimmune encephalitis. Okay, now I'm going to shift gears here and talk about some of our antibody work. Early on in my postdoc, my PI at UCSF, Joe DeRisi, I knew he was very interested in infectious disease, and so I hadn't really talked to him about my interest in autoimmune neurology, but it turned out that he wanted to work in that area as well, and so that was some real serendipity, and so as we were recruiting these mystery cases and sometimes finding wild infections, but oftentimes not, it was clear that it'd be really helpful to kind of have additional platforms to do antibody discovery. And so in addition to kind of the tried and true methods like rodent brain tissue staining, immunoprecipitation, mass spec, we also wanted to have a scalable platform that preferably would have a sequencing output, because we did a lot of sequencing, had a high signal to noise ratio. And so the platform that we settled on was phage immunoprecipitation sequencing, or FIPSeq, also called programmable phage display. And that was first developed in Steve Elledge's lab here with Ben Larman. And so we took a page from their book and developed a bit of an expanded human peptidome library, but identical concept in the sense that we computationally designed 49 amino acid peptides that overlapped each other by 50% and span all the protein coding regions of the human genome. We had oligonucleotides synthesized and cloned them into bacteriophage. You can see in the cartoon here, so that you can have in solution, just in a small volume of liquid, billions of phage, each of which are displaying on their surface one human peptide. Then you can take that library, incubate it with a patient sample like spinal fluid, let the antibodies bind to their cognate antigen, do an immunoprecipitation with protein AG beads to pull out antibodies in any phage that they've bound. And then we can do 2 or 3 or even 4 rounds of this to really narrow and heighten the signal. And then we just sequence the DNA from the immunoprecipitated phage, and we get back peptide counts. So we can look for enriched peptides above a variety of background controls. And so we've published a number of studies in this. We identified an antigen and a paraneoplastic syndrome in men with testicular cancer and a rhombencephalitis. A few years ago, we had these 2 studies were led by Callie Mandelbaum, incredibly talented postdoc who now is at lab at Yale. The second study was a rare pediatric syndrome called ROHAD. This was a collaboration with Leslie Benson and Mark Gorman, which she found the antigen ZSCAN1, interestingly, has no rodent ortholog. And so even though there had been a paraneoplastic etiology suspected in these children, when Dalmo and others had done tissue staining with spinal fluids from these kids, they'd never seen a staining pattern. So this antigen helped explain why, because it's just not present in rodents. But I'll tell you a couple of these stories. So one on MS, and then another one that's not here, but around a B12 story that we've been excited about. And so the MS work was led by Gavin Soa and Colin Zemechnik in collaboration with Mitch Walleen, and we were able to, Mitch enabled us to get access to this DoD serum repository, which is, I think people are familiar with, made famous by Alberto Scherio's group here, does really great work on, proving the link between EBV infection and subsequent development of MS. So we were able to access the same biobank, and we got 250 cases and 250 controls. And for each case and for each control, we were able to get 2 serum samples. So one sample came about, on average, 5 years before the person was developed their first MS clinical symptoms. And then the second sample came on average, a year or 2 after. And then the controls were time matched and matched on many demographic features as well. And so we ran those 1,000 samples on the FIPSeq platform. I should say too, we collaborated with Ahmed Abdelhak, who measured NFL on these samples. You can see even in the pre-symptomatic MS samples, that NFL levels, again, on average 5 years before clinical presentation were elevated, and even more markedly elevated, of course, after diagnosis. And so this is the giant heat map of kind of what Colin and Gavin found. And you can see there's a whole lot of blue, but in the MS population, there are some hot areas, and there's a number of them, but not in all the MS cases. About 10%, it turns out. So we'll zoom in here. So each column is a patient, each row is a peptide. If it's red or getting red, that means that peptide is enriched above the background. And you can see there's a number of peptides in this kind of 10% cluster of patients that are enriched. So one interpretation is that they have multiple autoantibodies. It turns out they actually have one. But that antibody actually is targeting a motif, the serine arginine repeat motif, that just happens to be very promiscuous in human protein. So it's present in a variety of human proteins. And so what initially looked quite complex is actually a single antigen, again, that's highly specific, but not particularly sensitive for MS and even as a pre-symptomatic biomarker for MS. It turns out that this motif has been reported in smaller MS, not pre-symptomatic, but in smaller MS cohorts. And you won't be surprised to learn that this amino acid sequence is also present in some non-human proteins, including a couple of Epstein-Barr virus proteins. So we think that there's a cross-reactivity here. What was interesting and what we still need to do more work to follow up on is Ahmed went back and broke down the NFL measurements based on whether someone was positive for this particular antibody or not. And in the patients with this immune cluster antibody, they even pre-symptomatically had a significantly elevated NFL relative to their seronegative pre-symptomatic MS colleagues. So, and that persisted even after diagnosis. So we think it's a predictive biomarker, but it's still unclear whether it could be prognostic in the sense that maybe these patients could have more aggressive disease, but we don't know that yet. We wanted to validate that, all these findings, and so we turned to a local incident MS cohort called Origins at UCSF and found, again, the same 10% of patients, both in spinal fluid and in blood, had this immune cluster signature. And then more unpublished work that we've subsequently been doing, this led by Krista McCutcheon, is developing a really kind of optimized Luminex assay that will allow us to probe more cohorts. So it turns out that because it's such a promiscuous motif, that initially we developed a multiplex assay, which included a few both viral and human proteins that have this same motif, which was it's an effective assay, but of course, the more analytes you include, you're going to affect specificity. And so Krista's done a lot of work, especially this was a key experiment where she used a tool called the Beacon to isolate antigen-specific memory B cells from a seropositive patient. I won't go into all the details here, but suffice it to say, starting with 39,000 memory B cells that she expanded to over 500,000, and then assayed 20,000 of them individually for whether they were secreting antibodies against this antigen. She found two B cells. Picked those two B cells. We sequenced the B cell receptors from those B cells. They were the same. And she made a monoclonal antibody that she's now has used to develop kind of an optimized Luminex diagnostic platform, both with a consensus peptide that to have a single analyte as a serologic tool. And so with that, we are looking now in MS familial cohorts to see, it's kind of why is this 10% number always coming up? Could there be a genetic predisposition to developing this particular humoral immune response? We don't know, but we're looking at familial cohorts and next month in a twin cohort. Also, we're kind of following the same playbook with some EBV-specific antigens in the hopes that maybe we could come up with a collection of highly specific, but maybe individually insensitive antigens that collectively might help us better diagnose MS. And then, and also with these monoclonals, we can put those into mice to see whether there are any pathogenic effects. Because again, this is a biomarker. We have no idea what role, if any, it plays in kind of the MS disease process. Make sure. Okay. So last vignette. This is kind of more on the encephalitis antibody discovery side. And to anyone in the room who's sent us mystery samples and you haven't heard back from us for a while in terms of whatever happened with that sample, this is a plug for saying just be patient. This was a case that we enrolled back in 2015, and it took a really talented neurology resident, John Pluvinage, whose picture will show up in a second, to make some sense of it. So I'll tell you this story. This is a woman, a middle-aged woman who presented to close colleague Jeff Gelfand's clinic back in 2014. She had a really profound ataxic syndrome and had this very unusual brain MRI where you can see these very symmetric T2 hyperintensities in the dorsal aspect of the brainstem extending into the middle cerebellar peduncles. And it was a bizarre case, which Dr. Gelfand sees many. And as part of his thorough workup, he didn't make a clear-cut diagnosis, but he found that the patient had lupus. And so she went into this grab bag of neuropsychiatric lupus cases, and he empirically immune suppressed her. She had some response, but over the years, and he still follows her in clinic, she continues to have some ataxia, also has some mild cognitive difficulties. And as you can imagine, like any good neurologist, Jeff checked her B12 levels many, many times, and they were always normal. But John came along and looked at the FIP-seq data from this patient, and he specifically had an eye in a number of lupus patients he was looking at, was looking for enriched peptides from cell surface proteins. So one of the knocks on FIP-seq is because you're displaying peptides and not whole proteins, you don't do a great job of picking up antibodies to conformational epitopes. So for instance, it's very insensitive for picking up NMDA receptor antibodies. But he thought, well, let's just, if we're going to find a function blocking antibody, let's just start there. And so what he got excited about was that in replicate, this patient had antibodies to a cell surface protein, and more interestingly, to a protein called CD320, which it turns out is the transcobalamin receptor. So it's the receptor that takes up B12, complex B12 into a cell. There's a long, wonderful history of kind of the history and discovery of, vitamin B12 deficiency, much of that occurring here in Boston. I think, at least as a resident, we were regaled stories where Marty Samuels and Alan Roper about. And so I don't think anyone in the room will argue that this might be an interesting lead. And so just from some publicly available data sets, John just looked at which cell type CD320 is expressed. Turns out it's highly expressed in endothelial cells, and then in particular, endothelial cells in the brain and in the testes. This is from another data set, again, from the brain, different brain cell types. And again, it's very highly expressed specifically in endothelial cells. So first, he wanted to validate that this was a true hit. So like in many antibody discovery papers, he did a cell-based overexpression assay of CD320 and showed that there was good co-localization with patient sample staining and a flag tag antibody. But then he dug a little deeper to see, again, because he wanted to really see whether this antibody might be pathogenic. And so he's interested about the specifically cell surface staining. So normally these cell-based assays use permeabilized cells. And in this case, whether the cell was expressing CD320 or not, there was staining, which is, you might get concerned about in terms of, whether this is a truly specific hit. But then we have to remember this patient has lupus. So she's got multiple other autoantibodies targeting the inside of the cell. And so he also looked at live cells where you're only going to see cell surface staining, and that was entirely accounted for by CD320 expression. The next question was, again, if the antibody is function blocking, presumably it should, and you see cell surface staining, it should be targeting the extracellular domain of the receptor. And one of the curse of FIPSEQ is that it's peptides, not whole proteins, but an advantage is that if specific peptides are enriched, you can pretty quickly localize where in the protein these antibodies are binding. And it turned out happily that this is the whole extracellular domain of CD320. This is the transmembrane domain. And the enriched peptides, not just in this patient, but in every patient we've looked at, are right here as a part of the extracellular domain. So that was also exciting. But then the next question is, does it actually block function? And so there is a holotranscobalamin kind of fluorescence-based uptake assay with live cells. And so John incubated these cells with the patient's spinal fluid to see whether the B12 uptake was affected by exposure to the antibodies in the spinal fluid. And he got more detailed than that. So he saw that just with kind of whole CSF, the uptake was blocked to a large degree. But then if he specifically depleted not just all antibodies from the spinal fluid, but specifically antibodies that bound to CD320, he could rescue the phenotype. So it was really looked like it was the CD320 specific antibodies in the spinal fluid that were inhibiting B12 uptake. And he did some more, then the question is, how does it inhibit it? He did some confocal microscopy suggesting that with exposure to antibody that the receptor is internalized. But then the big question is, well, who cares, right? So if the thought here is not that the antibody itself makes you sick, but that maybe because the receptor is expressed at the blood-brain barrier, if it's blocking function, blocking uptake, maybe people have a peripheral normal B12, but a low central B12 level. And so the big question was, well, what's the CSF B12 measurement? And in this case, in the index case, again, the B12 level in the blood was normal, and it's the same as controls, but in the CSF was essentially undetectable. So that really kind of drove it home that it looked like this person really had a compartmentalized B12 deficiency. Then John looked through we have a large FIPSEQ database. He looked through there and found there were actually some other patients that we had missed who similarly had antibodies targeting CD320 in this exact same region of the protein, validated all those cases with the CBA, showed that they were all inhibitory in this B12 uptake assay. And then we looked to see, well, what were these cases? So one was a patient from Mayo, from Owen Flanagan. He'd sent a few years before. And we asked Owen to get some more clinical information. And I forgot to ask him for the MRI, but he looked, thankfully, and was excited when he saw the MRI that the patient had a very similar kind of T2 hyperintensities, very symmetric. And as radiology, it suggested maybe metabolic picture. There was another patient who had been UCSF case who was diagnosed with a, quote, COVID myelopathy. So it was an elderly man, got COVID and presented with a profound myelopathic picture and had this really dramatic kind of tract-specific change in his MRI that looks a lot like subacute combined degeneration. And so many of these cases kind of fit with what we would think about clinically, radiologically with the B12 deficiency. We even went a step further and sent the spinal fluid to Norway to get MMA levels measured. And 4 of the 7 cases had not just low B12, but also elevated MMA in the spinal fluid, even though it was normal in the blood. So this is kind of John's working model. That patients can have these antibodies and be asymptomatic. We have detected this in healthy controls, but that over some period of time, people which is still undefined, and at some antibody titer still undefined, they can develop a centralized B12 deficiency, and then over again some other unclear period of time become more symptomatic. And just as an update on this, and I put these couple slides in here for Dr. Venna, because I'll get to that in a sec. But John's just been looking in our cohort at all our myelopathy patients. So they're about, again, this is not population representative. These are just referrals of difficult cases. But over the years, even though we're mostly focused on encephalitis, we've of course collected many patients with unusual spinal cord syndromes too. And so John found 37 patients, 18 of whom have, had subsequently gotten a diagnosis for their spinal cord disease, but about half still were, quote, idiopathic. And there have been a few more that have come in. So we're up to 26 that have idiopathic myelopathy. And then he compared these to 19 randomly selected age and sex match other neurologic disease controls. And in half of these idiopathic myelopathy patients, he's detected these antibodies, and all of those patients have low CSF B12. Again, one of those cases I've already shown you here, the COVID myelopathy case, but these are some additional. So 2/3 of the patients have dorsolateral column abnormalities. And I know Dr. Venn is very interested in this. We see patients with advanced HIV who can have a vacuolar myelopathy. It's kind of one of the cardinal, albeit rare, neurologic complications of advanced HIV. And two of the cases here were patients with CD4, with HIV and very low CD4 counts who presented with a pretty aggressive myelopathic syndrome. And you can see these pretty clear cut, again, dorsolateral column abnormalities, which again, we've called vacuolar myelopathy, but we haven't fully understood kind of the underlying pathophysiology. Okay, so I hope kind of, this last part is highlighted that we really can learn quite a bit from these very rare patients, both in terms, improving clinical care, but learning more about the underlying biology. We're doing more and more of the monoclonal antibody isolation. John's done that for the CD320 antibodies. I think those can be really useful tools, both to model disease, but also for diagnostic assay development, and even potentially for antigen-specific therapeutics like anti-idiotype antibodies. So I will stop there and just I've tried to highlight people along the way, but this takes an enormous effort, both at UCSF, my close collaborators, Joe DeRisi, Sam Pleasure, Steve Hauser, many others, and of course, all the members of my lab, and many outside collaborators. And then again, a big plug to the wonderful mentors here. I think just very excited for all the residents that you get to be part of this program, and it's just a real treasure. So, and of course, thank you to the patients. So thank you.
What are your thoughts? Their future program seems
Yeah.
like the follow-up from the serum to the CSF.
There we go. From the serum to the CSF was so steep that even very super therapeutic serum concentrations probably are not adequate. What about actual direct infusion to the CSF and a Meyer reservoir or something like that?
Yeah. So yeah, you're not letting me glide over that, which is good. Yeah, because it's challenging. So when MNGS was a research assay, if we found something, it wasn't real clinically, right? And so we would always, but we could whisper to the doctor, the treating doctor, say, hey, we found this weird virus. You should probably contact the CDC or someone and test for weird virus X. And then once it was validated in a clinical test, it was real and you could make clinical decisions. And so one way that, so this is all not real yet clinically, because one would hope like, okay, the antibody testing is research, but you could just send a clinical CSF B12 lab. And if that's low, who cares what the research antibody testing shows? Turns out there is no clinical CSF B12 lab either. So there's no way to kind of launder this into reality in the clinical world. And so, that said, B12 is not particularly toxic. And so anecdotally, there have been cases, like in the initial patient, with Jeff Galfan, where she did get super therapeutic, oral doses of B12 and actually got a repeat spinal fluid test. And what was fascinating was that in her case, at least, of course, her peripheral B12 level went up. And at the same slope, so did her CSF B12. The ratio between the two was still very abnormal, but it went up enough to get her in. And again, subjectively, she felt like she had cognitive improvements. But so I'm hand waving, just saying that there are anecdotal reports of people who have had some benefit, but I think first we need to kind of get this into like clinical grade testing and then do a more rigorous evaluation of different treatments. So yeah, but yeah.
You could maybe even think about a plasma exchange first and then a and see if that ratio changes.
Yeah,
based on
that's a great
presence of
point.
the
Yeah,
antibody.
Yeah,
But I was curious about the motif antibody,
yeah.
the genetic predisposition. Did you look for HLA restrictions, and maybe the 1501s are the ones who are reacting
Yeah.
even before they develop any signs of MS? Maybe you
Yeah,
could
it's a
screen their
great-
family members.
Yeah, so unfortunately, we didn't have HLA data on the giant DoD cohort. We do have it for the Origins cohort, but it's too small. It's underpowered. So Jill Hollenbach works with us, looked at that, but she warned me before, during, and after that analysis that we didn't have enough data, and she was right. So basically, we don't know yet, but that's where doing these, looking at these twin samples that are coming and familial cohorts, I hope we get a better sense of that. But right now, we don't have a clear-cut HLA association. But that, again, we just haven't been set up to find it yet. So.
Well, Michael, of course, thanks for coming back, for your visit, and for just a display of magnificent work over your career. It's just, I think I speak for all of us. We're so proud that you trained here and you went to somewhat of a sister institution and is really making a revolution in neuro-ID, in neuroinflammatory disorders. So a couple questions. The first, whether the myelopathy that you're seeing with the CD, with the antibody, whether is a progressive myelopathy or is a idiopathic, kind of a one and done type of a situation. And the other question is a little more broad, right? In the sense of using the beta genomic or the peptide strategy on these huge databases like the Department of Defense to really try to make a link of virus and neurodegenerative disease that we hear so much.
Yeah.
We often feel like we got to have a breakthrough.
Yeah. Well, thank you for that. I think it's a great question. It's too preliminary right now, but for the MS samples, I didn't get into this, but we also, just like the Alleged group, like we have a pan-viral antibody tool. So of course we're looking at these same samples with regard to that assay. But we haven't done it for in neurodegenerative disorders. But I think it's a fascinating question, and we're starting to think about that, at least, just with these the vaccination studies and things that I think we need to look. We've also, this is very developmental, but it may go nowhere, but in the patients with the TDP-43 diseases, like where we now know there's a lot of misspliced RNAs, and probably, some of those get made into, abnormal proteins. There's at least an outside chance that some of those might serve as neoantigens and provoke a humoral immune response. And so we've been working with Bill Seeley we made a different phage library that's not the human peptidome, it's not viruses, it's actually displaying all those potential misspliced proteins that have been cataloged with TDP43, just to see in the spinal fluid of those patients, can you detect an antibody response? So John Pluvinage is working on that as well. So that's a total flyer. I don't know where it's going to go, but yeah, I think, yeah, the nice thing is it's a bear to kind of get FIPSEQ up and running, but once you kind of have it going, then it's pretty plug and play. You can kind of make bespoke libraries to look at specific questions relatively quickly.
And in the phenotype of the B12 deficiency.
Yeah. So very progressive in a number. Yeah. So like I was just looking back last night at a couple of HIV cases, and those are older cases too. And they were quite, those patients progressed to being wheelchair bound over 8-9 month period. So, yeah. So, and yes, but then, in the COVID myelopathy case, that was quite acute. I don't think that COVID triggered it. I think probably he had these pre-existing antibodies, and then in the stress of a acute viral infection became more symptomatic. But so some of these patients have presented pretty fulminantly, but some are more kind of subacute to chronic, but it is progressive. Yeah.
I have another question for you, Michael. The metabolic angle is interesting to me. If you could block a B12 transport, what about other things? Folate, thyroid, anything else that needs to come in? Since you have the FIPSEQ data, do you have any other cases that kind of fit this model?
Yeah, so it's a great question. There was a report a few years ago, it was in New England Journal, of folate receptor autoantibodies, and I'm learning that there's kind of like every part of medicine, there's a whole kind of B12 subculture. They have their own meetings, and it's just amazing. It's so John's getting really into that. It turns out my division chief, Ari Green, his dad, Ralph Green, he's spent his whole career on B12. So he's been one of our advisors. Anyway, those antibodies were described. There was a diagnostic test, but it's not, I don't know it's not as widely available as it used to be. So we haven't detected with our tool, haven't detected those in the cohorts we've looked at. But again, I don't want to oversell FIPSEQ. This may be the one time we find a cell surface receptor antibody using this assay. So I think it definitely speaks to the fact that you need to complement this with things like yeast display, other whole protein display methods if you want to find those other types of antibodies.
That New England Journal was autism. There have been other papers, schizophrenia, tic disorder.
Yeah.
Have you looked at any of those populations?
A little. We have looked a bit in autism, and we haven't found a lot there. Yeah. So.
Thanks for the great talk. I'm just curious, you mentioned a little bit the yeast display. So, the FIPSEQ being linear peptides versus conformational epitopes. What's kind of your big vision for these undiagnosed cohorts of how you could combine the two methods for antibody discovery?
Yeah, I think, in these high likelihood cases, how, broadly defined, if someone has oligoclonal bands or staining or, or just clinically fits and, has a therapeutic response to immune suppression. In those, I feel like we need to just bring everything to bear, the challenge is that, there are obviously a few labs in the world that work on these things, but not all the labs have all the tools. So I think either we need to, we have had, the Kelch story, was a wonderful collaboration with Mayo. There's a different paraneoplastic biomarker that we identified with small cell lung cancer that case just sat on the shelf for almost 10 years now, but it was a single case. We didn't, anyway, we have recently collaborated with Jerome Honorat, who found 6 more cases. So I think we need to do a lot more kind of collaborative work, both to share samples, because these are all really rare conditions, and also just share techniques, because it's hard to kind of have in terms of funding and kind of technical expertise, to kind of have everything under one roof. We're trying to do that, but it's a lot. So I think ideally, there would be some way to share kind of these cases, both to be able to look for cousin, you find one patient and you don't, is it a snowflake? What does it mean? But when you find 2, 3, 4 that look very similar, then you get excited, but also to, if we don't find something by FIPSI, can we send it to a colleague to look with their assay as well? So, but because each of these platforms have very distinct advantages and disadvantages.
And do you think the better approach is to test? I think the thing that we've struggled with is, like, for instance, seronegative NMO.
Yeah.
It's probably 10 things. Like, we're trying to be like, how can we curate a cohort that's as similar as possible? Sarcoid has a similar issue of, maybe it's just a bunch of diseases that are granulomatous. Do you think that doing the approach of just screening all patients with a myelopathy is the better approach? Or should we try to say, in the B12 case, like, should we say, okay, let's combine all of the dorsal column, like, subacute-like cases?
It certainly the latter. I think if it hits, the story will make sense much more quickly. If you already have a clinically coherent group of patients, but at the same time, like I showed you, like we do both. We definitely screen cohorts like MS or sarcoid, et cetera. But we've also, really benefited from just not waiting for a cohort to accumulate and just looking at wild individual cases too. So I think, and that's where, something like FIPSI, again, it has disadvantages, but once you get it going, it's pretty quick and cheap to run. And you use just a few microliters of the CSF. So there's not a lot of harm in just peaking. You may not have the context to understand the finding right away, but we keep a large database, so we keep revisiting it over and over again. So, great. All right. Thank you, everyone. Appreciate it.